Dear students, in this module, I'm going to elaborate on the quaternary structures of the proteins. The quaternary structure, or simply the 4' structure of a protein, describes how a polypeptide chain talks with another polypeptide chain. The 4' structures of the quaternary structures are formed when different polypeptide chains they come together via hydrogen bonds, electrostatic interactions, and other types of interactions as well. An important thing that you should consider for this is that there are multimeric proteins and monomeric proteins. So if you have a monomeric protein, then the tertiary structure will describe the entire protein structure. But if you have a multimeric protein in which different polypeptides they come together and make a structure, then the quaternary structure is the complete structure for that protein. So, as I just mentioned, the proteins comprising of a single chain of amino acids, they can have a monomeric structure or the tertiary structure, while for the polypeptide chains which are acting together to make a structure, then they will give rise to a quaternary structure, and that will be the overall structure of that protein. To give you an example, here you have a quaternary structure, and you can identify, if you look closely, the yellow portion describes several alpha helices coming together in the form of a tertiary structure, and then they are interacting with this green alpha helices in order to give rise to a quaternary structure. And moreover, you see these blue alpha helices talking with the orange alpha helices and also making a multimeric polypeptide. Also, if you look at the cyan alpha helices, they are talking with the green alpha helices and they are holding entire structure together. So therefore, by simply coming together and holding each other, these monomeric polypeptide chains have given rise to a multimeric protein. So this describes the quaternary structure for this protein. So this will be the overall structure for this single protein now, instead of the tertiary structure that we discussed earlier. So in conclusion, the 4' structures are formed as a result of hydrogen bond formations, covalent bond formations such as the disulfide bridges, and electrostatic interactions, of course the hydrophobic interactions as well. So these interactions, they force the monomeric polypeptides to take form and take shape as a single protein. The underlying philosophy is that the 1' structure is less stable. The 2' structure, that is the alpha helices, beta sheets, they are more stable, but the tertiary structure is even more stable, wherein the alpha helices, beta sheets, coils and loops, they come together, while the 4' structure or the quaternary structure is the most stable protein structure that you have. All the proteins, they are aiming for evolving from the 1' structure to the quaternary or the 4' structure, and the underlying principle is their increased stability.